Hello dear viewers, welcome to the another informative video on multiple dissolution tests. So this video will be an interesting video, and in this video we will see why some of the monographs in the pharmacopoeias enlist the multiple dissolution tests, and what are the requirements for the product if there are multiple dissolution tests. So we will start from little bit basic about the dissolution. See, the dissolution is a performance test for the formulation, and it gives the understanding on how the product will behave inside the body. That is, the dissolution is the in vitro test done to ensure or predict the in vitro behavior, in vivo behavior of the formulation from the in vitro data. If your dissolution is meeting the specification in vitro, that means you can predict and ensure that the bioavailability will be there and there will not be significant changes in the bioavailability with the specific batch. The dissolution is a formulation dependent test. It is not like assay or impurity testing. It is very specific to the formulation. For example, you are making a formulation of, let's say, XYZ API. So assay limit will be more or less similar for all the formulations of that API, but dissolution test will vary. Then IVIVC describes the relationship between the in vitro property of the dosage form and the relevant in vivo response. Now see, there is a relationship between the dissolution and the in vivo behavior of the formulation. So that correlation is the relationship between the in vitro and in vivo response. So that is called as IVIVC. Then the dissolution method, parameters and the specifications are finalized based on the clinical studies, bioavailability and the bioequivalence studies. Now here we have to understand that whenever the new product is made, for example, you are working on the new type of tablets that is with specific release or specific uh, release kinetics with the old API. The formulation is new, the API is old. So you have made a formulation with some criteria for the bioavailability or the bioequivalence. Now the dissolution will be very specific for this type of formulation, and based on the clinical studies and IVIVC if available, the specifications will be finalized. So dissolution method, parameters and the specifications are finalized based on the clinical studies, bioavailability and the bioequivalence studies. Also, many of the time the shelf life studies are taken into consideration while giving the dissolution parameters and the specifications. Then the generic products can be manufactured by various means with different drug substances, variations in the formulation designs, and these products will be or may be bioequivalent to the reference formulations. The different formulation may have different behavior in dissolution and different disintegration and drug release mechanisms, but they may be bioequivalent. For example, one of the applicant is doing matrix tablet for extended release or sustained release or prolonged release, and the other manufacturer is doing a monolithic type of or coated type of pellets. So the tablet release will be different, but this product will be bioequivalent. The QC method or the monograph method may or may not give the true and biorelevance or in vivo prediction. Many of the time the QC method is adopted, and this QC method is finalized based on the product characteristics based on the API solubility criteria, but it is not the necessary thing that QC method will give you biorelevance or in vivo prediction. Also, generally the finalized QC method by the innovator formulations are given for monograph inclusion. So these QC methods may or may not give the true picture about the biorelevance. Then the formulations with different release mechanisms and rates may be bioequivalent to the same reference formulation. For example, if applicant A is using amorphous type of API and applicant B is using crystalline type of API, the polymorphs are different, but these both applicants are showing the bioequivalence to the same reference formulation. Then these two different applicants are using different APIs, so their formulation will be different and their dissolution specifications also will be different. Then the factors affecting the dissolution, mainly whenever variations are made, that time the API form affects the dissolution. Polymorphic form, solvent form, hydrate form, these all form of the same API or same drug substance may have different solubility and release kinetics. Then formulations can be made by using different drug substances forms with different solubilities and the permeabilities. API PSD also have a great impact on the dissolution. Different PSD API may give different release rates, but these all may be made bioequivalent with some formulation designs. Like if you are taking amorphous form, that amorphous form will be more soluble and crystalline form will be less soluble. Then solvent form and hydrate form may give you different solubilities and the different release rates, but may be bioequivalent. Then another is the solubility and the BCS class. BCS class 1 and 3 are highly soluble. BCS class 2 and 4 are poorly soluble. Dissolution can be achieved by different technologies like micronization, use of solubilizers, milling techniques, solid dispersion technologies, and use of some complexing agents also like cyclodextrins are there. So if different manufacturers are using different technologies, but they are giving the bioequivalent formulation or the formulation which is giving the good type of bioavailability and bioequivalence, that time their formulations will be different in the dissolution, but these formulations may be bioequivalent. Then the formulation design. So formulation design affects the drug release and the dissolution. So immediate release tablet will be similar to like IR suspension or solution filled soft gelatin capsules or granules. These all can be bioequivalent with different release rate and the formulation designs based on the drug solubility and the permeability. If the applicant is using immediate release tablet and another one is making the soft gelatin capsules, so the release rates and dissolution will be different, but these may be bioequivalent. Then modified release formulation can be made by different technologies like matrix tablet using the polymer, then osmotic tablets with the use of osmogen and the coating, and coated pellets filled into the capsules. All these formulation design will give the modified release formulation or extended release type of formulation, but these all will be the bioequivalent type of formulations. So the same dissolution method cannot be used for different formulation designs. Then there are more than one method to make the formulation. This is very important to understand that there is not only one way to make the formulation. There are various types of formulation designs and there are various ways to make the formulations. Then hybrid type of applications, which are also known as 505(b)(2) type, and the non-infringing type of formulations will be bioequivalent to the reference formulation with different release rate and the dissolution properties. Now multiple test in the USP. So if the generic formulations are made and these are being bioequivalent and these are being approved into the United States, that time these applicants will apply for the dissolution test inclusion into the USP. So the multiple test will be there in the USP for dissolution, drug release and the disintegration. The dissolution, drug release and disintegration test in the USP monograph are the tests. These are approved by the US FDA for products marketed in the United States. So this same case we may be with other type of pharmacopoeias or may not be. But specifically for USP, these all tests can be included and these tests are formulation dependent. So the single dissolution test may not be suitable for all products covered by the USP monograph. Then there are some examples like pantoprazole DR tablets are there and USP provides 5 different dissolution tests for pantoprazole DR tablets. And the applicant should include the test number in the label or leaflet, that is also known as patient information leaflet or PIL, if the test is other than the test number 1 is used. So this is the US market specific type of requirement and you may find number of products with such kind of the labels. If the product does not meet any of the tests in the monograph but the product is bioequivalent, then the product specific and discriminative method is required to be developed. Now if there are multiple tests into the USP for dissolution testing but still your product or applicant's product is not meeting to the dissolution specification with that test, then applicant can or should make the new method and which is required to be discriminatory and then that method can be included into the USP after the approval of the product. Then coming to the discriminatory method, this is the method which is able to detect the meaningful changes in the composition, manufacturing process or shelf life or age of the formulation sometimes. The new dissolution method can be submitted for inclusion into the USP as per the submission guidelines given by the USP. Then the dissolution procedure submitted to USP must include the sufficient details related to the critical test parameters such as medium, apparatus, sampling point and the tolerances, and also the quantitative method which are necessary to successfully perform the procedure and the result evaluation. Then justification for an unusual dissolution conditions should be provided as well. For example, you are using paddle apparatus and if you want to run that paddle at 100 rpm or 150 rpm or 120 rpm, then that will be little bit unusual type of dissolution condition. Or if you are going to use 2 liters of the dissolution media, then it will be a unusual dissolution condition which is very rare, and if it is there, justification should be available to use that unusual dissolution condition. Otherwise, the mild dissolution conditions like USP apparatus 1 basket with 100 rpm speed and paddle with 50 rpm or 75 rpm can be used. Then the complete details related to the method of quantitation, chromatographic, spectrophotometric or other analytical techniques must be provided. Here are some examples given that USP monograph for metformin hydrochloride extended release tablet contains about 15 tests for dissolution testing. And this is why, because the extended release profile can be achieved by different technologies. And all these products will be bioequivalent, will be approved by the US FDA, and applicant will submit the data for the dissolution test or new dissolution test to be included into the USP. So these are 15 dissolution tests. Another is USP monograph for diclofenac sodium extended release tablets. This contains about 5 different tests for dissolution testing. USP monograph for pantoprazole sodium delayed release tablets contains around 5 different tests for dissolution testing. And the example of labeling. If the product complies to this test, the labeling indicates that the product meets USP dissolution test 5. This is the example. And the reference for this presentation is taken from this reference, which is from the Dissolution Submission Guideline for Chemical Medicines, supporting information for dissolution drug release disintegration test in USP monograph. So if you download this article, you can get the information easily and you will have in-depth understanding of the multiple dissolution test in the USP. Then you might have a question that whether only the extended release or prolonged release or the delayed release type of formulations have different taste, and is it applicable to the immediate release type of formulations? So yes, for immediate release formulations also some different tests are given. If the formulation contains different salt form or different polymorphic form, that time the formulation will behave different in the different dissolution media and the dissolution conditions. So for that, the different dissolution test will be there and multiple dissolution test will be there into the monograph. And if the formulation is not in the monograph, that time the applicant have to develop or should develop a discriminatory dissolution method and submit in the application. So this is regarding the multiple dissolution test. I hope you might have got a good type of information out of this video, and we will meet you soon with such kind of informative and interesting video. Till then, take care, keep watching Pharma Learning in-depth videos, and please do like, share and subscribe, share to your friends. Thank you.